Hello, everyone. So, as you can tell, I apparently did a typo with the slides. However, we're having to roll with it because that's the kind of life we're having right now. Anyway, today we're going to be talking about molecular genetics, recombinant DNA, and genomic technology. So, my name is Shaylee Boger, and this will be for biochemistry. Um, 2 seconds. So the first thing we're going to talk about today is the biomedical importance. Um, so why does what we're going to be talking about, molecular genetics, actually matter? Um, so then it helps with personalized medicine, which um, I am very intimately aware with. I actually just had a presentation that I spoke at in April about the future of biotech and how AI can help us. And one of the things we did say was it would be leaning more towards personalized medicine. For me, for an example, I have Pompe disease, and there are 2 genome therapies that are available. I mean, 2 enzyme replacement therapies that are available that I cannot take because I'm allergic to corn. No one could have ever thought about having someone diagnosed with a corn allergy and Pompe disease when they were deciding this. So that. But an example, the person I was talking to, they were able to take a biopsy of his dad's cancer, and they were able to test it against different types of chemo, and they found that the first line chemo defense would not actually work against his dad's type of cancer. So it's really interesting. Another thing we could talk about is CRISPR. That's been in the news in the biotech industry recently because there was someone cured of a rare disease at CHOP. And just because I need to share it with someone, so I did just get a loaner sort of kind of computer to finish my exams because unfortunately I left my house this morning at 6 o'clock and I won't be back and my computer stopped working. So I had my old high school Chromebook, which is not supported, my mom's old Mac, and it's at iOS 10.13, you need 10.15 to support honor lock. So someone did let me borrow their Chrome, but it was let me borrow their Windows, but it's completely set to factory settings. And it's updating, and it's at 0%. So we're just hoping and praying that that happens, but worst case, I will email this to you. Anyway, I just needed to share that with someone. Anyway, so back to our conversation. Recombinant DNA technology involves a few seconds. It involves isolation and manipulation of DNA to make chimeric molecules. So the overarching is an end-to-end joining of different sources of DNA. Molecules with both human and bacteria, different human molecules. So far, it's just a very unique situation. Um, so one of the things that is important is there's restriction enzyme cleave DNA chains at specific locations. And they are bacterial proteins that are used to cut the DNA at specific sequences. So we will explain more as we get further in. Each one is named after the bacterium in which they derived. I originally set this up in my original video, I actually drew this all out, but third, fourth time's the charm. So we're going to just explain how we came up with the name SMA1. And I do call it SMA1, but not type 1 because that is a disease that has impacted a lot of people. So for an example, if we're going by the naming technology, S comes from Sierra, which is the genus, and then MA comes from the species, which is Marquez Atkins. Just a fun fact that bacteria originally causes UTI and pneumonias. Then the 1 in Roman numerals, or the I, is the designation of this. It is the designation of the strain, which means it was the first SMA1 gene that was used for restriction enzymes. And each cut is either blunt end or overlapping. Within overlapping, there's both sticky and cohesive ends. Sticky ends are more useful when constructing hybrid or chimeric DNA molecules. Moving on, hey, we're at 68%. Moving on to restriction maps. A restriction map is a map that allows a person to see representation of specific locations of a specific sites that restriction enzymes can be used. For an example, each one of these places is where a restriction enzyme can be used. And the designation is BAM or BGL or whatever. So as you can see, we did it with a lot with ones, but there's 2s, 3s, 5s, etc. So restriction enzymes, endonucleases, recombinase, and DNA ligaments are used to engineer and prepare chimeric DNA molecules. Sticky and cohesive ends are technically easy to cut. However, they require a lot of special techniques and expensive lab equipment. Some sticky ends may reconnect with themselves, which is the main reason why they prefer blunt end, is because you don't want to make it cut and then just make it reconnect with itself. Another thing with the sticky ends is you have to cut them at very specific locations, and what happens if they're not in the location you want. So blunt ends are directly litigated, so they're cut, but it isn't directional. And in order to solve the above problem, the T4 enzyme DNA, because they're not directional, in order to solve that problem, the T4 enzyme DNA ligaments are used, or RE reconstruction sites can be added to the DNA fragment with PCR. And then real world, restriction maps are helpful for CRISPR, as we did already talk about with the baby with the chop. And then I will say we are skipping a decent part of the chapter, which is a very interesting part of the chapter. However, I did think Gail did not want to be bored for the rest of the day with me just lecturing and lecturing, because I did choose this because it does impact me, because I do have Pompe disease. Recumbent DNA technology and disease. So normal gene variations are polymorphism and copy number variations. Polymorphism is the normal variation of DNA in a population. So for an example, there's only about 2% of a genome is part of your exome, and that is a majority of where your disease-causing situations are. However, it's also where all the other things are just where life happens. There is a large part of DNA we share like bananas. Copy number variations is the number of structural DNA changes between individuals. So for an example, there's just some random DNA changes. When you have your genome sequenced, they only look for disease-causing variants. They don't look for anything else, because there's some just random variants in there. Getting on to gene variation causing diseases, there's a couple of different ways. There's point mutations, which is, for an example, one base change. So my disease, Pompe, is an autosomal recessive. There's a change in the 32, and they all happen on the 17th chromosome, but there's a change in the 32, and they change the T to a G, and that causes a deficiency of the enzyme. And then there's deletion, which can be caused, which an example would be hemophilia B, because it is creating a deficiency of the factor 9. Transposition, you move one part of the DNA to another, and that could be completely minute. Yay. We're on step 2 of the other computer. Yay. Pedigree analysis. That is one thing that's important for people with genetic diseases, is to look at the family history and decide what genetic diseases might be there and what cannot be. Which leads us into our next thing, which is prenatal diagnosis. How does this help patients? How does this help researchers? Um, so you can be diagnosed prenatally through amniotic fluid, and it can be analyzed through southern blot transfer and PCR assays. And this helps both patients and researchers by allowing for an early diagnosis, which allows for patients to determine whether or not they carry the disease, but it also allows researchers to possibly, um, one of the things that biochemists help my disease so specifically is all of our labs are reactive. So after there's already a change that has happened, then you show a genetic, then your labs show an issue. For an example, our AST and ALT are impacted. However, all of that happened after muscle damage has occurred. Whereas right now, biochemists are looking to try and find a lab that would be helpful before the genetic mutations occur. And by diagnosing, I mean, not before the genetic mutations occur, before symptoms occur. And that's what prenatal diagnosis or newborn screening both help with. Is they allow researchers to have samples from people who are asymptomatic. Anyway, now we are going to switch to, now we're going to switch to, a few seconds. I am just going to, I did forget I added my questions. So let's go through and look at those. Let's see if I can. Make this happen. I really hope this is working. I really hope you're getting to see my terrible background. Anyway, I'm really just hoping. So question 1 would be, what is an example of biomedical importance? CRISPR, non-coding region, polymorphism, and naming theory, which are all things we talked about, but CRISPR's right, because it is actually why we look at the non-coding region and the polymorphism, and naming theory was just something that sounded fancy. How would we name the bacterium Thermus aquaticus? So just walking through this, we would take the T and then the AQ, and then if it's the one, I need to make sure that all of them are one, so that way then, so that way then no one would be confused. What is the type of DNA cut? Bond is obviously a chemistry bond. Fragment, you can do a litigation. It's not actually a cut. It's not. They use other things to create litigation, but it is not considered a type of DNA cutting. What is the type of gene variation? That could be disease causing. Polymorphism is not disease causing. Copy number variation, those are both non-disease causing. Deletion is disease causing, as for an example in the Pompe space, and then neutral, there are gene mutations that are neither bad nor good. And then why is genetics important when discussing biochemistry? And this is one of the things I think that ties in my entire discussion is research informed decision making, genetic controls all pathways, and all the above. So for an example, as we've been taking this semester and learning about the different biochemical pathways, almost all of them have a genetic background, something that codes genetic for that pathway. So anyway, I hope you have a great day. And then these are all of my references. I'm so sorry that this has been a interesting semester. I really I'm trying to find how to get back to the video. Anyway, hope you have a great day.